Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
Go above and beyond.
they become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. I have Vincent J. Maffei. He's in the Department of Microbiology, Immunology, and Parasitology at Louisiana State University in the Health Sciences Center. We're going to talk about alcohol and dysbiosis, intestinal problems because of it. I guess probably from alcoholics or maybe in other circumstances. But Vincent, thanks for coming.
Thanks, Richard. It's great to be here.
Yeah, tell me, if you would, about your research. What's it focused on?
Yeah, so I'm a former graduate student of the, as you said, the Microbiology, Immunology, and Parasitology department. I spent a lot of my time in the Comprehensive Alcohol HIV Research Center at LSU Health in New Orleans. And the CARC, as it's known, is tasked with the identification of the biomedical consequences of alcohol use in the context of an HIV infection. And so my particular project focused on investigating whether or not alcohol use is associated with accelerated aging in persons living with HIV. And so
it's really specific.
Yes.
How did you get into that? Why that?
Well, I think if you ask, you've probably spoken to a lot of scientists before, there's a lot of sort of meandering that kind of goes on when you're searching for a project. When I joined the MD PhD program at LSU in New Orleans, I didn't have a specific discipline that I wanted to study per se, but I did have a specific set of criteria that I kind of wanted to check off in my lab or in my project. Those were that I wanted to study informatics, bioinformatics. I wanted to learn how to code in Python and R, run statistical analyses, and then I wanted to apply it into a field that I felt had very broad applicability. And so my initial work as a graduate student was in the field of aging and aging biology related to dysbiosis. And we did some good work with that, where we were able to show that dysbiosis accompanies biological aging. And from there, I was interested in identifying maybe additional projects, even subpopulations of persons who are subjected to accelerated aging, and then to look at environmental exposures as they interact with the microbiome and potentially lead to changes in aging or changes in the rate at which someone ages.
Well, as a summary, what did you notice? How did microbiomes tend to change as people got older?
Yeah, so one of the primary measures of microbiome research that's done, especially from the standpoint of deep sequencing microorganisms in the gut, is the measures of diversity. And what we found was that as individuals age biologically, I say biologically as it stands in contrast chronologically. Chronological aging is simply getting older as time passes, but biological aging is getting older in such a way that your risk for future health problems increases. We have indices to measure one's biological age, so to speak. And what we found was that as people biologically aged, they tended to have less diverse microbiota in their gastrointestinal tract.
Oh, less diverse means in just terms of number of strains and species, or
number of unique organisms as identified by their 16S ribosomal RNA sequence.
Oh, what about, did you do any metabolomics or anything to see what the metabolic profile was?
Yeah, for this study, we didn't do true metabolomics. We did utilize a tool called PyCROSS, which is a tool to estimate or infer the genetic and genomic composition of samples as a function of biological aging. But we didn't actually measure directly, just because the scale of the study was relatively small, and at the time, the funding kind of wasn't really focused towards that. That sort of work is beautiful and something that I see myself getting into, down the road. It's a perfect project for a bioinformatician because, metabolomics, just like the microbiome data, you study thousands and thousands of features, and the goal is to, identify these collections of features that are important and potentially have meaningful use in the clinic.
So, all right. So, how did this turn to alcohol for and how it affects people's gut that have
Yeah.
HIV?
So that after completing that sort of early project in my work, we asked ourselves, what's next? What else is available? And it turned out that the Comprehensive Alcohol Research Center, had this ongoing study known as the New Orleans Alcohol and HIV Study. This study is a cohort, it's a prospective longitudinal study of about 365 human subjects. They all have HIV, and fecal samples are being collected from these subjects. And the goal was to identify whether or not, there are predictors of perhaps dysbiosis in these participants, and whether or not those measures of dysbiosis predict or related to, at least statistically related to, changes in outcomes, HIV-related outcomes, and in particular, how T cells age is one particular outcome.
Why is this even important?
People with, interestingly, HIV, these numbers were just updated in June, but there's a study out by Dr. Julia Marcus from Harvard and her team that shows that people with HIV, on average, they live a lifespan that's about 9 years shorter than the general population. This is actually much improved from about 20 years ago, where that lifespan gap was more on the order of 20 years or so. So as time has gone by, our ability to fight this virus essentially and preserve the lifespan has improved, but there's still a major gap, in terms of lifespan with people with HIV and compared to those without. In addition, what's interesting is that people with HIV tend to acquire health conditions, aging-related health conditions at an earlier rate. They tend to acquire it earlier in life compared to those in the general population. And we're talking about a gap in time to first comorbidity on the order of about 15 years. And so people with HIV, they, even despite control of their virus, there's still stuff that's going on in their bodies that's leading to, shortened lifespan, and in addition, more years that are unhealthy.
So are these people that are just drink the occasional drink, or are these alcoholics, or what's the profile of the cohort?
That's a great question. Yeah, so the question is, how much alcohol is harmful in people with HIV? Is it heavy drinkers that are the ones that are really driving these, potential phenomenon? And the truth is that there's some good work done by the Veterans Aging Cohort Study authors that show that, there's actually really no safe drinking level for people with HIV. In fact, those that are that do drink tend to have a higher risk of mortality per level of alcohol consumption compared to the general population. There's something about having HIV that alcohol use really just doesn't mix with it. And so this has stimulated a lot of research into identifying the different reasons as to why alcohol can lead to earlier mortality in people with HIV compared to those without.
You have a cohort of just regular folks that drink as well?
So for that arm of the study is actually ongoing. We initially had funds to essentially do an HIV only cohort, and now we've actually started this process of bringing in controls to look at the HIV negative cohort. Those that are also living within the New Orleans area but that don't have HIV. And so this study of ours really just looked at a wide spectrum of alcohol use. I guess our controls in this study would be those that have little to no alcohol use compared to those with some, compared to those with a lot.
So what are you noticing?
So we're finding, very interestingly, in fact, what started or inspired this line of inquiry was a pilot study done by our group, where this was done in a rhesus macaque model of simian immunodeficiency virus. This is the cousin of HIV. And these SIV-infected rhesus macaques, chronic binge alcohol administration was associated with an increase in the number of CD8 T cells that are expressing a senescent phenotype. So senescence in T cells is deleterious. Senescence is a state of essentially impaired replicative ability. And if you recall, it's replication, making copies of a cell's self, is an essential step in a immune reaction. When an immune cell interacts with a pathogen or some sort of antigen, and it registers it as foreign and needs to be removed, it makes several copies of itself. That's an essential step in an immune response. And these cells, they lose that ability. And this is thought to be a consequence of repeated and prolonged episodes of immune activity, right? So we're talking about people living with HIV. They have an infection that is unfortunately, not cured. It's something that they live with for their lifespan. It's something that's managed with drugs, but that virus lingers, and with it lingers chronic inflammation. And so it's thought that, people with HIV are sort of predisposed to this prolonged aging or this accelerated aging through this chronic low-level inflammation. In addition to that, alcohol use, we believe that alcohol use is exacerbating that phenomenon. Alcohol is a very inflammatory molecule, and in particular, its metabolite, acetaldehyde, is very inflammatory. And so, we contend that the inflammation associated with alcohol, and certainly chronic alcohol use, can lead to exacerbation of the phenomenon of T-cell aging or T-cell senescence. And,
yeah, but there's got to be a lot of studies on microbiome and alcohol use so they can inform you on what to look for.
Well,
what did you go in expecting to see and why?
So our main study was essentially had two arms to it. The first one was to try to see if we could validate or confirm some of the observations made in that animal study I just mentioned, where we would look at individual participants' alcohol use and see if it's associated with the emergence of senescent CD8 T cells, in their blood samples, and we were actually able to do that. We actually did find that there was a positive association between one's risk for an alcohol use disorder and the emergence of senescent CD8 T cells. And then as a follow-up, we asked, what might be mediating this mechanism, or what might be mediating this phenomenon? And the first thing that came to mind was dysbiosis, one, and then two, a second phenomenon known as gut leak. And so there's quite a bit of literature and this is outside the field of HIV, looking at just the effects of alcohol on the gastrointestinal tract. And there's a number of animal studies that have shown that when alcohol is administered to an animal, its gut barrier breaks down. The gut epithelium is designed to keep out, pathogens. That's essentially one role of the gut epithelium, but it's designed to keep those microbes out and keep them in the gut so that they don't find their way inside the host. Well, alcohol disrupts that. Alcohol, permits these microbes to migrate from, the lumen, the inside of the gut, and into the host tissues. And as you can imagine, that generates a very inflammatory response, both locally at the site of the gut, but also systemically. And so we came into this project, with the goal of identifying whether or not alcohol is leading to gut leak in our cohort, right? And so we also assayed markers of gut leak to see if we could, capture this phenomenon, at least in our cohort. And interestingly, that didn't pan out as strongly as we were expecting. And there's different reasons, why this might be the case. Of the three markers we looked at, essentially one of them was positively associated with alcohol use, but the other two were not. And we think this might be related to the fact that, in these animal studies, they're being fed, pretty high levels of alcohol, and then their guts, gut leak is essentially measured shortly after that administration. But in our cohort, our participants, they were required to not have had any alcohol consumed within the past 24 hours. And so this is telling us that potentially, this gut leak phenomenon may occur, during a state of intoxication, but it may not occur outside of that. So that particular hypothesis, potential explanation of why alcohol is leading to CD8 T cell senescence, didn't really pan out completely, I should say.
In your human cohort, you're telling people not to drink for 24 hours before they
show up.
Why? Why not look at
yeah, so that's a great question.
Right after you drink, 6 hours, 12 hours, a mini longitudinal study.
Yeah, that's a really good question. And, I misspoke earlier. We didn't have participants withhold for 24 hours. We had them withhold starting at midnight the night before a study visit. In fact, we actually had participants initiate a period of fasting at that time, so no food or alcohol at that time. And that's because we were assessing parallel questions on alcohol's association with metabolic syndrome and insulin resistance. And one of the objective measures of those that we collected was a fasting glucose level, which requires a period of fasting testing before you collect, a blood glucose level. So, but it is interesting to ask the question, how might the outcomes that we've discussed today, senescence, dysbiosis, gut leak, how might those have been a little different had we allowed participants to consume alcohol as they normally would have leading up to study visit? The question as to alcohol's acute effects on those outcomes would be, is an interesting one and would be more directly addressed in the setting of a controlled trial where participants could come to a very secure location, consume a fixed or controlled amount of alcohol, and then those outcomes could be, directly measured on site. And so that's, that would be a particularly interesting study to do. That's not one that we've planned for the near future. It's one that has been done before by other groups in other contexts. So it's interesting to talk about such a study nonetheless.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
Why don't you look at oral microbiome?
Well, we do. We actually have that. We actually collected those samples, and so that we certainly haven't excluded that analysis. For me, I was sort of the grad student on point for the microbiome project, and logistically, what worked the best was to really kind of work on the gut question. We have oral rinsates that we've collected from our patients. And so, I think down the road, perhaps the next grad student that comes through the lab could certainly, tackle that project and look at changes in the oropharyngeal microbiome and even look for just some connectivity between the oropharynx and the gut that might explain, maybe some of these gut changes are explained by some changes in the oropharynx, right? Maybe some of this dysbiosis begins in the oropharynx, right? And so
it would have to because you're drinking the alcohol, so it hits there first, and I'm sure it kills a vast number of the existing bacteria there and probably acidifies the mouth and does all kinds of stuff.
Yeah, and just to build off of that, oropharyngeal microbiome has to pass, the stomach. It's going to pass stomach acid. It's going to make it past the small intestine, which is, very harsh. Those are both very harsh environments. And maybe alcohol suppresses the, essential digestive functions so that those organisms are allowed to pass. Maybe alcohol lowers the gate, so to speak, on entry into the gastrointestinal tract.
So in this study, are you far enough along where you are seeing effects? Are you saying just, again, a reduction in diversity or what kind of
so what we found, we measured alcohol use 4 different ways. One of those was just estimating one's risk for an alcohol use disorder. This is based off of questionnaires that assess people's patterns and behaviors of drinking that befit a AUD or an alcohol use disorder diagnosis. We also measured alcohol on the measure of on the order of a lifetime. We had them sort of rate their broad use of alcohol during different decades of their life. We also had them report their use over the past 30 days. And then we measured a biomarker. We actually measured a biomarker of alcohol use that responds to alcohol use within about, I'd say in the past 2 weeks. And so it gives you some measure of alcohol exposure in the past 2 to 3 weeks. And we found that, interestingly enough, the biomarker was associated much more closely with dysbiosis than the others, which is interesting because our measure of risk for an alcohol use disorder was strongly associated with senescence. Interestingly, this biomarker was associated with dysbiosis and senescence. So we were expecting to see some symmetry between these measures of alcohol use, senescence, and dysbiosis, but we didn't see that. And so that sort of speaks to this potential ephemerality of this phenomenon, where maybe alcohol use in the short term can lead to changes in the microbiome, which then correlate with changes in senescence. But perhaps these changes in the microbiome may not persist in such a way that you would be able to pick up those changes by just identifying those that have an AUD.
All right. So you're gauging senescence by what? The action of T cells that they're apoptosing more, or how do you gauge senescence there?
Yeah, so that's a good question. So for this study, this sort of early study, we looked at cell surface markers that were indicative of senescence. And so in particular, the CD28 molecule that is normally expressed on T cells, on senescent T cells, that molecule is missing or downregulated. And so that, in conjunction with other markers, we use to identify a senescent CD8 T cell. And down the road, in fact, our lab is working on, actual assays to measure true senescence, to actually take a sample of cells from a patient, stimulate them, and check their response. Are they able to replicate? Are they able to mount an effective immune attack or immune response to, let's say, some sort of lab-derived foreign cell? Right, you can put in these exogenous cells that are not from the patient and measure how well they're killed off by the patient's immune cells. And so those are more direct measures of senescence, right? Because you're looking at function, you're looking at replication. And so we are building those techniques. Those are not trivial assays. They're very expensive and they take a lot of troubleshooting. But at this stage, this is we're in a very early stage in our research center. So this is how you get to that point. You do studies that focus more on markers. Markers are easier to collect. Markers, you can do them on a larger scale usually. And so this is how you build up to work like that.
I mean, what's the end goal here? I would think you would tell people that have AIDS don't drink.
Yeah, right.
That's probably not going to change.
Yeah, I mean, it's much more complicated than just educating them on the harms of alcohol use, because alcohol use is, it's a chronic relapsing addiction, right? And so it's much more, alcohol leads to sort of a rewiring of reward systems. So it's hard for people to quit, right? Just recognizing that not everyone is going to quit. The end game here is to identify probiotics, prebiotics, some sort of microbiota intervention that could rectify at least this component or this source of inflammation in people with HIV, such that they could, hopefully experience a longer lifespan, a longer period of healthy years as opposed to unhealthy years. Again, those numbers I quoted earlier. The goal is to narrow the lifespan gap and narrow the gap in incidence of first comorbidity with the general population. So just like any other translational scientist, you start sort of broadly and you sort of work your way in. The goal is to identify, more precise mechanisms that can then be leveraged in a therapeutic modality.
Well, again, how much is understood about just alcohol use in general and how it affects the microbiome?
And so, right. So in terms of the work done in animals, there's quite a bit of work. I mean, it's hard to make a statement of how complete that work is, right? How much more has to be done. There's an incredible amount of research that spans decades on the effects of alcohol use in humans, the effects of alcohol use in animals. I'll say that right now, the attention in one of the big projects in the alcohol field is how do you measure alcohol directly as the person is drinking. There's actually been some projects that have been funded to develop wearable technology to actually measure one's consumption on a real-time basis. That way, you have an objective measure of consumption that follows the patient everywhere they go. That way you don't have to rely on self-report, right? People, it can be hard sometimes to self-report accurately how much you've consumed over a certain period of time. So, the tension right now is, how do we improve our measures of alcohol consumption and do it in such a way that doesn't require any special thinking, that's not as error prone. And from there, make better inferences about how alcohol influences health. And so until you get to that point, the sample sizes for these studies will have to be very large because there's going to be a lot of variability in how consumption is reported. But if you can get a tool that's precise, you have the ability to potentially utilize a smaller sample size to accomplish the same sort of goal as you would otherwise.
Okay. I mean, so preliminary data just shows a reduction in the number of species of bacteria in the gut. Is there any correlation between the reduction in a given species, less bifidobacteria and, health consequences? I mean, what conclusions do you draw?
So one surprising finding was that as people had greater recent alcohol use as measured by this biomarker, it was found that there is a co-abundant group of Prevotella bacteria within the Prevotella genus, and then other Prevotelaceae members within the Prevotelaceae family. This co-abundant group of bacteria, interestingly, was positively associated with recent alcohol consumption, but also positively associated with CD8 T cell senescence. And then to kind of connect them even further, we did a statistical mediation analysis that just sort of looks at this sort of co-movement of these three phenomena, and we found a significant result where statistically the co-abundance of these Prevotella organisms statistically mediates the relationship between alcohol use and T cell senescence. And what's interesting about Prevotella is that there's actually been a lot of attention, a lot of good data out there on the role of Prevotella in HIV infection. Prevotella has been known to be more abundant in persons living with HIV, and in particular, a subpopulation of people with HIV that are also men who have sex with men. So Prevotella has been sort of classified as a potential very interesting player in dysbiosis HIV biology. Another interesting finding from another group was that Prevotella, when it's incubated with infected CD4 T cells, that infection tends to be more productive. More of that infection seems to take into the cells. The cells seem to produce more of markers of HIV virus, and those cells are actually die off more quickly compared to incubation with other bacteria in the gut, like Bacteroides or Bifido, like you were saying. So Prevotella is, right now a sort of a hot topic in the HIV microbiome literature, and we're excited to sort of contribute to that because we've looked at an environmental exposure that's very common in people with HIV that may be regulating Prevotella.
Well, I just realized too that anyone that has HIV is going to be on who knows how many drugs.
Right.
So I would think that that would radically change the microbiome and probably radically change how alcohol interfaces with those drugs too. There's a lot of contraindications.
Yeah.
So don't drink if you take X.
Yeah.
Is that being looked at, or is this all just? I mean,
yeah.
It sounds like there's a lot of confounding going on.
Oh yeah, there's a lot of work that still needs to be done. The trick with these studies, these multidimensional studies, is that there's so many things that need to be controlled for and looked at, and you do this as well as you can, sort of incrementally. You don't just publish one study and just call it a day. We try to keep building on what we've learned about the microbiome and other factors that influence alcohol use, and try to incorporate those in subsequent studies. In our study, we didn't necessarily control for which ART regimen they were on. However, we did control for whether or not they were adhering very well to it or not. So we separated those subjects statistically and made sure that we accounted for the effect of people that that are adherent to ART religiously compared to those that are having more trouble doing so. And ART, is antiretroviral therapy. These are small molecules that, by design are supposed to interact with RNA reverse transcriptase. That is, at least some of those drugs interact with the RNA reverse transcriptase. So these drugs naturally interact with DNA, and bacteria harbor DNA, they harbor RNA. Bacteria are, strange organisms. They have, they produce many different types of polymerases, et cetera. So there's reason to believe, my point is that there's reason to believe that ART could certainly interact with these bacteria, could alter them in ways that are not really expected. And so that's another important study, is to sort of fish out the differences between the microbiome that are explained simply by consumption of different ART regimens.
Is there research in that? HIV infected people and what happens to their microbiome in general, with or without drugs, longitudinally?
Yeah, interestingly, some of the early work has showed that people with HIV that start, so in general, people with HIV tend to have a less diverse microbiome. When you add in ART, when you give them ART, there's a replenishment. It seems like those that are actually taking their drugs and are achieving, suppressed viral load, their diversity goes back up. And so there seems to be, this is sort of contributed to this message that diversity in general, at least in the gut, higher diversity is better, lower diversity is sort of worse. This sort of contributes to that field in that kind of way, that as you start to target the HIV infection and suppress it, you get a healthier, presumably a healthier gut.
I just don't know if diversity in terms of 16S is really going to tell you much. I mean, there's diversity of function again, but metabolomics and proteomics and stuff. So I mean, since there's redundancy in the gut in terms of which species can do X, Y, or Z. These
are early studies, right? And so you start looking in these very complex studies, you start looking at markers first, right? And that's what 16S studies are. You're looking at a marker gene. This is just a region of a taxonomically informative gene that then gives you information about the origin, or perhaps the phylogenetic origin of the organism. And then from there, it's sort of inferred more or less that the function is different. Obviously, there's a lot of assumptions that are made when you perform a 16S study. Claim that there's dysbiosis, and then claim that there's a change in function, right? So yeah, I mean, at the end of the day, there are better ways to assess dysbiosis, right? One way that our lab is working on is to take fecal samples and then actually incubate them under anaerobic conditions, and then collect a supernatant from these fecal samples, and then use the supernatants, which are presumably containing metabolites from the bacteria, filter out the bacteria out of these supernatants, and then treat different cell populations to look for example, changes in senescence, right? Or activation of a HIV genome within a latent T cell reservoir, right? And so this is another way to potentially get at the question of dysbiosis. Unfortunately, the microbiome is so vast and so complex. It's difficult to think of a model or a model where you can take the entire community, test it without, interrupting it in some kind of way. It's sort of the holy grail of microbiome research is to develop some kind of model that you can work with this gut microbiome exactly how it is in a human without actually using a human or an animal. So that's a major sort of line of work. And at this point, we're just sort of tangentially getting at testing the true function of the microbiome at this stage in the research. But in the future, I certainly look forward to better methods that could be developed to answer these questions, because you're absolutely right.
Well, if you're going to make a supernatant of fecal matter, I mean, you may just be filtering out bacteria, but what about the phagome? It may not just be metabolites. I mean, there could be all kinds of other stuff in there.
Yeah, absolutely. The phagome is super interesting. That was something that I had originally proposed to do in my graduate work, but we ultimately sort of decided against it just for logistics. But the phagome is fascinating. I mean, when you filter out bacteria, you're not filtering out viral particles, right? Those viruses are way too small to be captured by these filters. Yeah, I mean, one way to, I guess depending on whether or not you see a virus as a living thing or not, as abiotic or biotic, you could even consider it as a metabolite in a way, in that, or really just more broadly as a byproduct of bacteria, right? Because these phages, they grow inside bacteria, they're released into the environment. And absolutely, phages could certainly play a massive role in how bacteria compete with each other, how they work with each other to dominate the gut space for resources and nutrients. It's entirely possible. We talked about the oropharynx and how alcohol can potentially lead to changes in the gut as a function of as a function of what bacteria are in the oropharynx. Alcohol is a stressor, right? Alcohol stresses bacteria. For some bacteria, alcohol can be used as a nutrient. For others, it's a stressor. And so one thing that phages and other viruses that have integrated into host cells, what they often do in response to a stressor is to replicate. They want to make more copies of themselves before it's too late, right? So it's totally reasonable to believe that alcohol could be inducing maybe a stress response and inducing phage release, which then leads to massive shifts in the microbiome. Absolutely.
Yeah, well, there's a lot to figure out. So what's the path forward for the research in the near term, the next year or so?
Yeah, like I said, the main goal is to create more functional assays to really get at the things that we intend to study. Functional assays for senescence, functional assays for dysbiosis. We're trying to continue this work in our cohort. Our study is actually a longitudinal study. So we're doing the same sample collection at a later date, and we're going to look at longitudinal changes in alcohol use and dysbiosis and senescence to see, is there a temporal relationship between changes in alcohol, changes in the makeup of the gut that might correspond or explain some of the differences in T cell senescence. And so the longitudinal arm of this study is ongoing, and that's something to look forward to.
Well, very good. Vincent, what's the best way for people to find out more about your work?
Yeah, so I've provided some links to the Comprehensive Alcohol Research Center. You simply just type in, the Comprehensive Alcohol Research Center, LSU Health Sciences in Google, and it'll probably be your first hit. That's the best place to start. And on that page, you'll see the different lines of inquiry that have been established by our research center. And so from there, you can get in touch with us and maybe express some interest, maybe express some criticism, what have you.
Yeah, I guess, people need a stiff drink when they read through the research, right?
Yeah, absolutely.
No, well, that's great, Vince. Well, thanks for coming on the podcast. I appreciate it.
Thanks, Richard, for the invitation.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.